Namaskar, welcome to Whitetail. Here we will explore the science behind RK-251, how it works, what researchers have discovered so far, how it differs from conventional chemotherapy.
Imagine a world where cancer treatment no longer inflicts harm on healthy tissues while effectively targeting malignant cells. Enter RK-251, a groundbreaking advancement in cancer therapy that seeks to revolutionize how we approach treatment. By functioning in an inactive state until it encounters a specific chemical environment, this innovative drug takes a step toward minimizing the side effects commonly associated with traditional chemotherapy. This paradigm shift allows for a more precise targeting mechanism, sparking hope among both patients and healthcare providers. Before we delve deeper into RK-251, it's critical to understand why conventional chemotherapy often comes with a myriad of side effects. Chemotherapy's typical mode of action is the destruction of rapidly dividing cells, a characteristic that not only applies to cancer cells but also to several normal cells, such as those in hair follicles and the digestive tract. As a result, patients frequently endure unpleasant side effects during their battle against cancer, from nausea to hair loss. While chemotherapy remains a pivotal treatment known for saving lives, the quest for alternatives that offer a more refined approach is paramount. This is where RK-251 makes its mark. Developed through a collaborative effort between the Institute of Advanced Study in Science and Technology, IASST, and IIT Guwahati, RK-251 has emerged as more than a mere chemotherapy drug. It is termed a ROS-responsive fluorogenic prodrug, a name that may sound complex, yet encapsulates a brilliant concept. Essentially, a prodrug requires specific chemical or biological conditions for activation, allowing for therapeutic components to be unleashed precisely where needed. This activation mechanism is like possessing a medicine delivery system that remains dormant until it reaches its intended target, thereby maximizing efficacy while minimizing collateral damage. RK-251 brings forth a glimmer of hope, not just as a potential chemotherapy alternative, but as a stepping stone toward a future of smarter cancer treatments. The active engagement of the drug, based on the cancer cell's unique chemical environment, sets a foundation for more personalized medicine. Researchers and oncologists are now optimistic that through diligent exploration of RK-251, they may discover an effective means of combating cancers with significantly reduced side effects, yielding greater quality of life for countless patients battling this relentless disease. These cells produce reactive molecules known as reactive oxygen species, or ROS. Typically elevated in cancerous environments, these ROS levels present a unique opportunity for scientists to exploit this difference in cellular chemistry. Imagine using a cancer cell's own environment to trigger a potent anticancer drug, ensuring that the treatment is more targeted and effective. Enter RK-251, a cleverly engineered compound designed to respond specifically to ROS. The underlying principle is elegantly simple. When RK-251 encounters high levels of ROS, it chemically activates, releasing an anticancer molecule called NBDHEX. This molecule operates by inhibiting GSTP1, an enzyme often overexpressed in various cancers that aids in evading cell death and contributes to drug resistance. The beauty of this method lies in its precision. Rather than dispersing an active drug molecule indiscriminately, RK-251 carefully controls the release of the therapeutic agent right where it's needed most, inside the cancer cells. Additionally, RK-251 has a remarkable feature that sets it apart in the field of drug development: the ability to produce a near-infrared fluorescent response upon activation. This allows researchers to visually observe the activation process in real time, essentially witnessing the drug being activated and the subsequent release of the anticancer agent. This fluorogenic approach to drug delivery opens new avenues for monitoring treatment efficacy and safety, providing insights that were previously difficult to glean. However, cautious optimism is warranted when interpreting these findings. The next phase of research utilized laboratory models of triple-negative breast cancer, focusing specifically on MDA-MB231 cells. The results were striking. RK-251 exhibited potent anticancer activity against these cancer cells while sparing non-malignant cells. Moreover, experiments involving developing zebrafish embryos demonstrated the anticipated fluorescent response without any acute toxicity or abnormal development. While these results are promising and indicate a potential breakthrough in cancer treatment, it is essential to understand the context of these studies. They are, after all, preclinical research, indicating that RK-251 has not yet been tested or approved for human use. Therefore, while the prospect of using the chemistry of cancer cells to our advantage is exhilarating, we must wait for further testing before concluding whether RK-251 can safely treat cancer in patients. In the landscape of cancer research, the inquiry surrounding RK-251 presents a pivotal point of discussion. Can it truly replace chemotherapy? Currently, the answer remains a resounding no. Despite the excitement surrounding RK-251, making such a claim at this stage would be hasty and scientifically unfounded. The road ahead is infused with questions waiting for answers, questions fundamentally crucial to patients' safety and treatment efficacy. Before RK-251 can transition from experimental status to legitimate therapy, a thorough exploration of its safety profile in humans is paramount. Determining the appropriate dosage, understanding how the body metabolizes the compound, and assessing its capacity to effectively penetrate tumor sites must be achieved. It is equally vital to discern whether various cancer types will respond differently and to predict any potential side effects that may arise. A critical overarching question lingers: does RK-251 actually improve patient outcomes? Answering these questions requires extensive preclinical work followed by meticulous human clinical trials, thereby ensuring that the potential benefits are matched by proven safety and effectiveness. History has shown that while many compounds dazzle in laboratory settings, they often do not secure the transition into approved medicines. Thus, it is essential to celebrate the advancement of research without allowing our hopes to transform into unreasonable expectations of instantaneous miraculous cures. Currently, RK-251 remains within the realm of experimental findings and cannot, at this point, be considered a substitute for established chemotherapy. The importance of RK-251 lies not solely in the molecule itself, but in the innovative strategy it represents. Rather than merely focusing on finding chemicals to eradicate cancer, researchers are shifting their attention towards understanding the unique environment of cancer itself. This nuanced perspective aims to exploit the differences within the cancer ecosystem. RK-251 seeks to harness elevated levels of reactive oxygen species, ROS, as a mechanism to activate its anticancer properties. Should this strategy prove effective and safe for human use, it may herald a new era of tailored cancer treatments. It stands as a noteworthy achievement in scientific inquiry. Having birthed a potentially transformative prodrug that operates through the ROS pathway, it invites attention and consideration. However, it is imperative to recognize that the advancement of science progresses through a rigorous path. Discovery leads to testing, validation, safety assessments, and ultimately, the necessity of clinical trials that yield robust evidence. The journey of RK-251 has commenced, and its future rests in careful exploration.